Day 1.
Alright, so...
Hey guys.
This time.
So, why don't you guys take a look at this?
The cell museum, you know that has the highest level of security out of all of the cells in this organism, right?
Exactly. That's why we have to plan this much better than every single other time. Cell wall, you're in charge of getting us past the first security check. This means letting Golgi in. Golgi, you're going to be doing the actual sealing. So you'll go in and transfer the artifact to me, where I'll seal it and I'll store it in my usual place. Got it?
Got it.
And we'll be able to sell this for 500 grand.
500 grand? What exactly are we stealing?
We're stealing a painting. It's a lost painting, and it's called De Planta Cell, which translates into English to the plant cell. It's by Plastic Van Gogh.
Alright, I got it, but I gotta go. We can talk later.
Okay, see you guys. Day 2. So, hey, what have you got for me?
Alright, so I, as a Golgi Apparatus, will deal with the transportation of everything. So recently, I received a loop from our outside supplier, the endoplasmic reticulum. I'm going to send that loop to the cameras in the cell museum to shut them down, similar to how I will send modified lipids and proteins to different parts of the cell in order to help it function. Now, once that's done, that's going to give me time to steal the actual painting. I'm going to come in with the fake painting and replace the real one with the fake painting. Once I get the real painting, I'm going to send it to you, Central Vacuole, and you can store it, alright?
What about you?
Well, the cell wall, the rigid layer that lies outside the plasma membrane of plant and bacterial cells, will pretend to be a security guard at the cell museum. Because you guys can enter the museum through my pores. Because of my pores, I can allow the passage of water, ions, and other molecules. And because I am a plant cell wall, I am made up of cellulose embedded with a matrix of proteins and other carbohydrates. So that means I will be able to maintain the cell shape and keep a stiff barrier around the cell museum. And if anything goes wrong, then I'll be able to build a secondary cell wall, like the ones I make in some plants when the cell stops growing, that does not expand and that will strengthen the protection and the rigidity of the cell museum doors.
Okay, so are you guys ready to do this?
Alright, we got this.
Okay. 1, 2, 3.
Organelles!
See you guys tomorrow. Day 3.
This is the cell museum. Just follow the plan and don't listen.
Alright, got it.
Alright. Center vacuole? Alright, I got it. It's coming to you.
Okay, I can store it now.
Alright.
Day 4. Okay, you guys. So this is my place.
So this is your place, huh?
What is this place, Central Vacuole?
So this used to be a cell daycare center until I restored it and I turned it into my storage room. So when I first found it, it was full of all these disgusting bugs called enzyme pigments, and they would crawl around everywhere. It was disgusting. And whenever it rained, it would flood the entire building and make all these water molecules everywhere. And last but not least, it was obviously covered in waste and toxins. But then I restored it last summer. I also added this new cement thing called the tonoplast, and I added it to all the walls, which means that it'll maintain turgor pressure and it'll keep everything stable. That keeps everything from collapsing in on us. Let's go in. I think the dealer's waiting for us.